Welcome back to the deep dive. Today, we're really peeling back the layers on something, well, fundamental to how we exist, how we move, that honestly most of us rarely give a second thought to.
That's right.
We've got some incredible source material here, going deep into two brain structures that are just essential for, normal motor function, the cerebellum and the basal ganglia.
Yeah, absolutely essential. Because while the main command, the sort of go signal to move a muscle, might start up in the cerebral cortex, that big outer layer of the brain, these two structures, they're more like the crucial coplets, or maybe the engineering team.
Right.
The sources call them accessory systems. That's a key term. They can't by themselves make a muscle contract, but they work in association with the main motor systems. That's the important bit, to make sure movements are smooth, coordinated, happen at the right time, the right intensity.
Okay, so our mission, our goal for this deep dive is really to pull apart the details in this material. We want to look at the structures themselves, the circuits connecting them, their specific jobs, and importantly, what happens clinically when things go wrong.
Exactly.
We really want to get a handle on how these accessory systems shape purposeful movement, making it, effective.
And there's this fascinating paradox that sources point out right at the start. One of these, the cerebellum, historically, it was thought of as a silent area.
Silent? How so?
Well, if researchers stimulated it directly with electricity, the person didn't report feeling any conscious sensation, and you didn't see much obvious movement happen either, so
Yeah.
it seemed quiet.
Huh. Okay, but there's always a but in neuroscience, isn't there?
Always.
Yeah.
But if you lose cerebellar function, maybe through damage or disease, that's a completely different story. The sources are incredibly clear. Damage causes wildly abnormal movements.
So how can something that seems silent, that doesn't cause sensation or movement directly, be so vital? That just seems contradictory.
It really does on the surface, but that's the core puzzle we need to unpack. Its job isn't about being the ignition switch for movement. It's more like the master craftsman, the finely tuned engineer working constantly behind the scenes.
Refining things.
Refining, sequencing, learning from mistakes, making corrections on the fly. It's all about the quality control and coordination. Let's start by really digging into the cerebellum first.
Okay, the cerebellum, this supposedly silent area, we heard why it got that name, that initial impression, but the sources immediately hit us with this counterpoint, take it away, and movement becomes profoundly abnormal. What kind of abnormality are we talking about? Is it weakness?
That's the crucial distinction right there. Its loss doesn't cause paralysis. You can still initiate muscle contractions, still generate force. The problem is coordination. The movement becomes clumsy, inefficient, jerky, profoundly uncoordinated.
Okay.
And this is especially true, the sources emphasize, for any movement that's rapid or requires quick changes, quick adjustment.
So things like running.
Yeah, absolutely. Or typing on a keyboard, playing a musical instrument like the piano, even just speaking clearly requires incredibly fast, coordinated muscle movements.
Right, the complexity of speech is amazing when you think about it.
It really is.
Yeah.
And these kinds of fast, skilled actions, they just fall apart without the cerebellum working properly. The coordination breaks down.
So its role isn't flipping the switch for movement, but somehow guiding it, steering it once it's started.
Precisely. That's a great way to put it. The sources explain that the cerebellum's primary job is to help sequence motor activities, monitor them as they happen, and make these instantaneous corrections. It's constantly running this feedback loop.
Okay, tell us more about that loop. What kind of information is flowing through it? What's it actually processing?
It's getting two critical streams of information simultaneously. Constantly. First, it gets continuous updates from the other motor control areas in the brain, primarily the cerebral cortex, but also brainstem areas. These updates tell it about the intended muscle contractions. Exactly. The motor plan. What muscles are supposed to contract? In what order? With what force? It knows what the brain wants to do next.
Okay, so it's got the blueprint. What's the second stream?
At the very same time, it's receiving incredibly detailed, continuous sensory feedback right from the periphery, from your body.
And the muscles themselves.
From sensors embedded in your muscles, yes, muscle spindles. Also from Golgi tendon organs in your tendons, from receptors in your joints, even tactile receptors in your skin. This stream reports the actual current state of your body parts.
Like telemetry data.
Exactly like telemetry. Their precise position, how fast they're moving, the rate of change of movement, what forces are acting on them, tendon tension, all that real-time physical data.
So it's got the plan from the cortex and the reality report from the body. And its job is to compare them.
That comparison is absolutely central to its function. The cerebellum constantly takes that detailed sensory feedback about what's actually happening in your limbs, your trunk, wherever, and compares it moment by moment against the motor commands, the plan that was sent out.
And if there's a mismatch, if the reality doesn't line up with the plan, say you didn't lift your foot quite high enough.
That's precisely where the corrective action kicks in. If the actual movement deviates from the intended trajectory, maybe a muscle contraction was a bit weak, or a joint angle is slightly off, or you encounter unexpected resistance, the cerebellum instantly calculates the error.
And does what with that error calculation?
It transmits rapid, subconscious corrective signals. These signals are sent back into the motor system,
Back to the cortex.
often back towards the cortex via the thalamus, or to brainstem motor nuclei like the red nucleus. These signals effectively modify the ongoing motor commands, adjusting the activation levels of specific agonist or antagonist muscles to nudge the movement back towards the original intention, closer to the plan.
And this is all happening in real time, fractions of a second, completely without us having to think, oops, lift foot higher.
Completely subconsciously, continuously, for almost every voluntary movement you make. But it's not just reacting to errors after they happen. Another really important detail from the sources is that the cerebellum also helps in planning the next movement in a rapid sequence.
Before the current one is even finished.
Exactly. Just a fraction of a second before the current movement element is completed, the cerebellum is already involved in setting up the parameters for the very next one. This anticipatory planning, this pre-calculation, is absolutely crucial for achieving smooth, fluid transitions between individual movements in a sequence.
Like when you're typing a word, each finger movement seems to flow seamlessly into the next without conscious thought for each transition.
That's a perfect example. That seamless flow relies heavily on this cerebellar anticipation. And what if, even with the correction and anticipation, the movement still isn't perfect? Especially when learning something new.
Does it just keep correcting?
It does, but it also learns. This is maybe one of its most amazing functions. The cerebellum learns from movement errors. The sources explain that the sensitivity of its internal neural circuits can actually change over time. Specifically, how its main output neurons, the Purkinje cells, respond to inputs gets modified based on the errors detected, that discrepancy between intention and outcome.
So practice really does make perfect, and the cerebellum is where that motor learning happens.
A huge part of it, yes. With practice, those circuit adjustments mean that future attempts at the same movement become progressively more precise, more accurate, conforming better and better to the original intention. It fine-tunes the motor program based on experience.
Wow. So it's a real-time comparator, an error corrector, an anticipatory planner, and a motor learning device, all operating below the radar of consciousness that is incredibly far from silent in its actual function.
Absolutely. The silent label really only applied to that very specific context of direct electrical stimulation and conscious sensation. Functionally, it's constantly buzzing with activity.
Okay, let's look at how its physical structure supports these incredibly complex roles. The sources go into quite a bit of detail on its anatomy and functional divisions.
Right. Anatomically, the cerebellum is described as having three main lobes: the anterior lobe, the posterior lobe, and the much smaller, older flocculonodular lobe down at the bottom. These are separated by deep fissures or grooves.
And that flocculonodular lobe, the oldest part.
Yes, phylogenetically the oldest. And its function aligns with that ancient role. It's very strongly linked to maintaining body equilibrium balance. It works hand in glove with the vestibular system in your inner ear.
Makes sense. Balance is pretty fundamental. But the sources also emphasize functional divisions that run sort of longitudinally along its length, particularly in the larger anterior and posterior lobes. These seem key to its different motor roles.
Yes, this longitudinal organization is critical for understanding its function. If you look at the cerebellum from the back, there is a narrow band running right down the middle. This is called the vermis.
The worm.
Exactly, from the Latin. This central vermis area primarily controls the muscle movements of the axial body. So think your trunk, neck, shoulders, and hips, core stability, and postural control.
The midline, controlling the midline of the body. Okay.
Right. Then, moving laterally outwards from the vermis on each side, you have the large cerebellar hemispheres. These hemispheres are themselves functionally subdivided. Immediately next to the vermis is the intermediate zone.
Intermediate zone controls what?
This zone controls the muscle contractions in the distal portions of your limbs. So primarily your hands, fingers, feet, and toes. The finer movements out at the ends of your arms and legs.
Okay, so vermis for the core and posture, intermediate zones for the more detailed movements of the hands and feet. Makes sense. What about the rest of the hemispheres, the outer parts?
That's the lateral zone. And this is the largest part, especially in humans. The sources make a really key distinction here. These large lateral zones seem to operate at a more abstract, higher level of motor control. They work very extensively with the cerebral cortex, particularly in the overall planning and timing of complex sequential motor movements.
Ah, so this is where that smooth sequencing, like in typing or speaking, really gets organized. And damage here causes those coordination breakdowns in rapid activities.
Precisely. The sources emphasize that without the proper function of these lateral zones, most discrete, rapid motor activities lose their appropriate timing and sequencing. They become clumsy, disjointed, uncoordinated. The fluid progression is lost.
Now, do these different zones have maps of the body like we see in the motor cortex or the sensory cortex? You know, a little representation of the body laid out on the surface.
That's another important point. The sources state that the vermis, axial body, and the intermediate zones, limb phase, do have topographical representations, yes, body maps. And significantly, these mapped areas receive sensory input from the corresponding body parts they represent. And they also get input from the corresponding topographical areas of the cerebral cortex and brainstem motor centers. And crucially, they send motor signals back via output pathways to influence those same topographical areas. It's a geographically specific loop.
A dedicated circuit for controlling specific body parts. What about the large lateral zones, the planning and timing areas? Do they have body maps too?
Here's where it differs, and it's really telling. The sources state that the large lateral zones do not have topographical body maps. Their connections are different. They receive input almost exclusively from the cerebral cortex.
Which parts of the cortex?
Specifically from the higher level planning and association areas, the premotor cortex, supplementary motor area, and importantly the somatosensory association areas and other sensory association areas, like in the parietal cortex. Areas involved in integrating information and formulating plans, not just executing commands.
Ah, okay. So they're wired into the parts of the cortex that are thinking about the bigger picture, the strategy, the sequence, the integration of senses, rather than the parts directly controlling individual muscle twitches.
Exactly. This pattern of connectivity, receiving input from cortical planning areas and sending output back to them via the thalamus, is perfectly suited for the lateral zone's role in anticipating, planning, and coordinating the timing of rapid sequential activities, helping the cortex basically choreograph the whole movement sequence.
It's like the cortex comes up with the concept, and the lateral cerebellum helps work out the detailed timing and flow.
That seems to be a good way to conceptualize it based on these connections. And structurally, the whole cerebellum, the sources describe it as this massive, highly folded sheet of neural tissue.
Yeah, like a cauliflower shape almost.
Right. Very convoluted. If you unfolded it, it would be surprisingly large. It consists of this outer layer, the cerebellar cortex, folded into ridges called folia. And nestled underneath this cortex, deep within the white matter, are the deep cerebellar nuclei.
And these deep nuclei sound critical.
They are absolutely critical. Because they are the primary source, the main output stations for almost all the signals leaving the cerebellum to influence the rest of the brain's motor systems.
Okay, let's trace that flow of information now, starting with the inputs. What are the major pathways bringing information into the cerebellum? The afferent pathways, as the sources call them.
There's a huge amount of information converging on the cerebellum. From other parts of the brain, one of the most important is the corticopontocerebellar pathway. This is a massive pathway. It originates widely from the cerebral motor cortex, the premotor cortex, and also the somatosensory cortex.
So key motor and sensory areas.
Yes. These fibers project down to nuclei in the pons, in the brainstem, the pontile nuclei. And from there, second-order neurons cross over and project primarily to the lateral divisions, the lateral zones, of the cerebellar hemispheres. And note the crossing over input from one cerebral hemisphere goes to the opposite cerebellar hemisphere.
Right, contralateral connection. So the main planning link from the cortex feeds directly into the cerebellum's main planning zones. Makes sense. What else comes from the brain?
There is also the crucial olivocerebellar tract. This originates from a structure in the medulla called the inferior olive. Its fibers, known as climbing fibers, project to all parts of the cerebellum.
All parts, so it's a very widespread input. What information does the inferior olive send?
The inferior olive itself seems to be an important integrating center. It receives inputs from the cerebral motor cortex, the basal ganglia, parts of the brainstem reticular formation, and also sensory information relayed from the spinal cord. So it's already combining multiple streams.
Right.
This pathway via the climbing fibers is thought to be especially important for motor learning, signaling errors, as we'll get into more detail later.
Okay, an input pathway, potentially signaling errors, coming from a place that integrates motor and sensory info. Interesting. Any others from the brainstem?
Yes. We also have vestibulocerebellar fibers. These carry equilibrium information directly from the vestibular apparatus, the balance organs in your inner ear, and also from the brainstem vestibular nuclei, which process that balance info. These fibers mostly terminate in the sloculonodular lobe and the associated deep nucleus, the vestigial nucleus.
Reinforcing that balance function connection.
Exactly. And finally, reticulocerebellar fibers come from the brainstem reticular formation and terminate mainly in the midline cerebellar areas, the vermis. This links the cerebellum into the brainstem systems for controlling posture and background muscle tone.
Okay, so input from the brain covers high-level cortical plans, integrated signals possibly related to errors, balance information, and basic postural control. What about that critical sensory feedback coming directly from the body itself via the spinal cord?
This is delivered extremely rapidly and directly. The two most important pathways described are the dorsal spinocerebellar tract and the ventral spinocerebellar tract.
Okay, dorsal and ventral spinocerebellar, what information do they carry? Let's start with the dorsal.
The dorsal spinocerebellar tract enters the cerebellum via the inferior cerebellar peduncle, one of the connecting stalks, and terminates mainly in the vermis and intermediate zones on the same side of the body for which the signals originated, ipsilateral.
And the information it carries.
It primarily carries signals from muscle spindles, those sensors telling about muscle length and rate of change, but it also includes information from Golgi tendon organs, signaling muscle tension, tactile receptors in the skin, and joint receptors. It's basically giving the cerebellum a high-fidelity, real-time report on the status of muscle contraction, tendon tension, the position and rate of movement of body parts, and any forces being applied to the body surface. It's the detailed feedback on the actual physical state.
The reality report we talked about earlier.
Precisely. Now, the ventral spinocerebellar tract is fascinatingly different. It enters via the superior cerebellar peduncle and, interestingly, terminates on both sides of the cerebellum. Bilateral projection.
Both sides, okay. And its information source is different too.
Yes. What's really key here is that it receives less direct input from those peripheral sensory receptors. Instead, the sources say it's excited mainly by the motor signals arriving at the anterior horns of the spinal cord.
The motor neurons in the spinal cord?
Exactly. It's monitoring the signals coming down from the brain via pathways like the corticospinal tract and the rubrospinal tract, as well as signals generated by the spinal cord's own internal pattern generators, like for walking.
So it's not reporting what the muscles did, but what the motor neurons were told to do.
That's the crucial interpretation. The ventral tract tells the cerebellum what motor commands actually reach the final common pathway, the anterior horn cells, regardless of whether the movement perfectly matched that command or not. The sources refer to this as the efference copy of the anterior horn motor drive. It's a copy of the outgoing command signal as it arrived at the spinal cord level.
Ah, I see. So the dorsal tract gives the actual result feedback, and the ventral tract gives the command sent feedback, or a copy of it.
Exactly. And having both allows the cerebellum to do that critical comparison. It can compare the intended command, efference copy via ventral tract, with the actual outcome, sensory feedback via dorsal tract, to detect errors and discrepancies much more effectively.
That's incredibly clever, and these pathways are fast.
Extremely fast. The sources mention conduction velocities up to 120 meters per second, making them among the fastest pathways in the entire central nervous system. This speed is absolutely necessary for the cerebellum to make those instantaneous, real-time corrections needed for rapid movements.
And the sources mention other peripheral inputs, too, relayed through different spinal pathways and brainstem structures, like the reticular formation and inferior olive again.
Yes, reinforcing this picture of the cerebellum as this central hub, constantly, voraciously collecting subconscious information about body movement, position, forces, and the commands being sent, all converging from multiple sources.
It synthesizes all this incredibly rich, dynamic data. Okay, now let's flip the coin. How does the cerebellum send signals out? The efferent pathways.
These pathways originate primarily from those deep cerebellar nuclei we mentioned earlier. The dentate nucleus, largest, associated with the lateral zones, the interposed nucleus, associated with the intermediate zones, and the vestigial nucleus, associated with the vermis. The vestibular nuclei in the brainstem also functions somewhat like deep nuclei for the flocculonodular lobe's output related to balance.
And these deep nuclei, they receive input from two places, right? Directly from the incoming afferent tracts and also from the cerebellar cortex above them.
That's exactly right. And this pattern leads to a really fundamental principle of how the cerebellum processes information. When an input signal arrives in the cerebellum, say via a mossy fiber or a climbing fiber, it splits.
Splits?
Yes. One branch goes directly to synapse on a neuron in one of the deep nuclei. This connection is excitatory. It gives the deep nucleus neuron a quick push.
Okay, direct excitation to the output nucleus.
The other branch of that same input fiber goes up into the overlying cerebellar cortex. It activates the circuitry within the cortex, eventually exciting the main output neuron of the cortex, the Purkinje cell. Now, the Purkinje cell's connection back down to that same deep nucleus is inhibitory.
Ah, so the input excites the deep nucleus directly, but then after a slight delay going through the cortex, it triggers inhibition of that same nucleus.
Precisely. The deep nucleus gets an initial excitatory kick, followed almost immediately a fraction of a second later, due to the processing time in the cortex by a precisely timed inhibitory signal from the Purkinje cell it's connected to.
Excitation followed by delayed inhibition, that seems like a core operational motif.
It is. Virtually all inputs ultimately cause this biphasic excitatory then inhibitory sequence in the deep nuclei. And the final output signals that leave the cerebellum to influence the rest of the motor system originate from these deep nuclei, whose firing is constantly being shaped by this interplay of direct excitation and delayed cortical inhibition. This mechanism is thought to be absolutely critical for the cerebellum's role in timing and damping movements, as we'll explore more when we look at the microcircuitry.
Okay, let's trace where these outputs go. What are the major efferent pathways from the different deep nuclei, starting with the vestigial nucleus from the vermis?
Right, the vestigial nuclei receive input mainly from the vermis, axial control, and parts of the flocculonodular balance. Their output signals project primarily to the medullary and pontile regions of the brainstem.
Targeting brainstem areas.
Yes, specifically connecting with the vestibular nuclei, further reinforcing balance control, and the reticular formation, involved in posture and background tone. So this pathway directly links the cerebellum's midline structures back to the brainstem systems, controlling equilibrium and posture.
Makes sense. Core and balance control output. What about the interposed nucleus, the output from the intermediate zones controlling distal limbs?
The interposed nucleus projects more widely. A major target is the thalamus, specifically the ventrolateral VL and ventroanterior VA nuclei. These thalamic nuclei then relay the signals up to the cerebral motor cortex.
Ah, a loop back to the cortex.
Yes, but the interposed nucleus also sends fibers to other targets, including other midline thalamic nuclei, parts of the basal ganglia, the red nucleus in the midbrain, which gives rise to the rubrospinal tract, important for limb control, and parts of the upper brainstem reticular formation.
Wow, that's quite a spread of connections. What's the overall job of this pathway?
The source identifies its main function as coordinating the reciprocal contractions of agonist and antagonist muscles, particularly in the distal limbs, the hands, fingers, thumbs. It's about the fine-tuned push and pull needed for precise manipulation and skilled movements of the extremities. This aligns perfectly with the intermediate zone's focus on distal limb control.
Okay. And finally, the big one, the dentate nucleus, the output channel for those large lateral zones involved in planning and sequencing. Where does it project?
The dentate nucleus, the largest of the deep nuclei, also projects primarily to the ventrolateral VL and ventroanterior VA nuclei of the thalamus. And from the thalamus, these signals are relayed predominantly back up to the cerebral cortex.
Which parts of the cortex? Back to the primary motor cortex.
Significantly, the return projection goes mainly back to the same cortical areas that projected to the lateral zones in the first place. That is, the premotor cortex, the supplementary motor area, and the association cortices.
Ah, so it completes that cortex cerebellum cortex loop specifically for the planning and association areas.
Exactly. It's a closed loop connecting the cortical planning centers with the cerebellar lateral zones and back again. This pathway is considered absolutely crucial for the cerebellum's role in helping the cortex coordinate sequential motor activities, particularly those that are rapid and complex. It reinforces the lateral zone's function in planning, timing, and ensuring smooth progression between movements.
These distinct output pathways really mirror the functional divisions we discussed earlier. Vermis vestigial for posture balance, intermediate interposed for limb coordination, lateral indentate for planning sequencing via the cortex. It's a very logical layout.
It is indeed. Now, shall we zoom in even further to that fundamental functional unit you mentioned, the one centered on a single Purkinje cell and its corresponding deep nuclear cell? The sources say there are something like 30 million of these units operating in parallel.
30 million, a massively parallel processor. Okay, let's dissect that core microcircuit.
Right. The basic functional unit, as depicted in the sources, involves one of those very large Purkinje cells in the cerebellar cortex and a corresponding neuron in one of the deep nuclei below it. Well, the deep nuclear cell is ultimately the output neuron for this specific unit, sending its axon out of the cerebellum.
And its activity is modulated by that balance of excitation and inhibition we talked about.
Precisely. The deep nuclear cell receives direct excitatory input from the afferent fibers coming into the cerebellum. Both types, the climbing fibers and the mossy fibers, send branches to excite it. But it receives its only inhibitory input from that single associated Purkinje cell located above it in the cortex.
Only inhibited by its Purkinje cell.
That's the described circuit. Normally, at rest, both the deep nuclear cells and the Purkinje cells are tonically active, firing continuously at moderate rates. The balance between the tonic excitatory input and the tonic Purkinje inhibition is such that the deep nucleus usually has a steady, moderate background firing rate. It's the changes in this balance that encode the output signal.
Okay, now let's look at the inputs arriving at the cerebellar cortex itself, the ones that eventually influence the Purkinje cell. The sources highlight two distinct types of input fibers, climbing fibers and mossy fibers. Let's start with the climbing fibers. They sound quite unique.
They really are. Climbing fibers originate only from the inferior olives down in the medulla. That's their sole source.
One source for all climbing fibers. Okay.
And each climbing fiber connects intensely with only a very small number of Purkinje cells, perhaps just 5 to 10. But the connection it makes with each Purkinje cell is incredibly powerful. It wraps around the Purkinje cell's dendrites like a vine, forming maybe 300 or so synaptic contacts.
Wow, 300 synapses from just one fiber onto one cell. That must have a huge effect.
It does. A single action potential arriving along a climbing fiber causes a massive prolonged depolarization and burst of firing in the Purkinje cell. This unique response is called a complex spike. It's a very strong, almost overwhelming signal for the Purkinje cell. And importantly, remember that these climbing fibers also send collateral branches to directly excite the deep nuclear cells before they even reach the cortex.
Okay, so climbing fibers originate only in inferior olive, connect powerfully to few Purkinje cells causing a complex spike, and also directly excite the deep nuclei. Very specific, very strong input. Now, what about the other type, the mossy fibers?
Mossy fibers represent all other afferent fibers entering the cerebellum. So these carry inputs from the cerebral cortex via the pons, the brainstem, and directly from the spinal cord, like the spinocerebellar tracts. They carry the bulk of the information about the motor plan and the sensory feedback.
So a much more diverse origin than climbing fibers. How do they connect?
Like climbing fibers, they also send collateral branches to directly excite the deep nuclear cells. That direct excitatory input is common to both types. However, within the cerebellar cortex, their connection pattern is very different. Mossy fibers synapse with a huge number of tiny interneurons called granule cells, located in the densely packed granule cell layer, the deepest layer of the cortex.
Granule cells, aren't those the most numerous neurons in the entire brain?
By far, billions of them. Each mossy fiber excites many granule cells. These tiny granule cells then send their axons upward into the molecular layer, the outermost layer of the cortex. These axons are extremely thin, less than a micron in diameter.
Okay.
Once in the molecular layer, each granule cell axon splits into two branches that run horizontally, parallel to the long axis of the cerebellar folds, or folia. These are the famous parallel fibers.
Parallel fibers, and there must be an immense number of them.
Absolutely astronomical. Because each mossy fiber excites many granule cells, and each granule cell axon gives rise to two parallel fibers running in opposite directions for a millimeter or two. There are literally millions upon millions of these parallel fibers packed into the molecular layer.
And these parallel fibers are what synapse onto the Purkinje cells.
Yes. The huge, fan-like dendritic trees of the Purkinje cells extend up into the molecular layer, oriented perpendicular to the parallel fibers. As the parallel fibers run past, they make synaptic contact with the spines on the Purkinje cell dendrites. Each Purkinje cell might receive input from tens of thousands, maybe even up to 200,000 different parallel fibers.
A massive convergence of input. But are these individual synapses strong, like the climbing fiber ones?
No. The sources describe these parallel fiber synapses as being relatively weak. Unlike the climbing fiber's powerful effect, a Purkinje cell typically requires the near simultaneous activation of a large number of parallel fibers, spatial and temporal summation, to be significantly excited and fire an action potential.
Okay, so climbing fibers give a strong, focused, complex spike, while mossy fibers, via the granule cell parallel fiber system, provide a much more widespread, diffuse input that requires summation to trigger a different kind of spike in the Purkinje cell.
Exactly. The action potential generated in a Purkinje cell by parallel fiber input is a more standard, brief spike, typically referred to as a simple spike. So, the Purkinje cell is constantly integrating these two types of signals: infrequent but powerful complex spikes from its climbing fiber, and much more frequent but weaker simple spikes driven by patterns of activity across many parallel fibers. And remember, the Purkinje cell's output, driven by these spikes, is always inhibitory onto its target deep nuclear cell.
Right, the Purkinje cell is the inhibitory bottleneck controlling the output from the deep nucleus. The sources also mention a couple of other cell types in the cortex: basket cells and stellate cells.
Yes, these are other types of inhibitory interneurons located within the molecular layer. They're also excited by the parallel fibers. Their axons, however, travel sideways, perpendicular to the parallel fibers, and synapse onto nearby Purkinje cells.
What's their function?
Their primary role is thought to be lateral inhibition. By being excited by a beam of active parallel fibers and then inhibiting Purkinje cells adjacent to the ones most strongly activated by that beam, they help to sharpen the signal. It enhances the contrast, making the response of the main target Purkinje cells stand out more clearly by suppressing the activity of their immediate neighbors. It's a common principle used throughout the nervous system for refining spatial patterns of activity.
Okay, sharpening the signal. So we have this intricate microcircuit. Incoming afferents excite the deep nucleus directly. They also activate the cortex either strongly via climbing fibers or diffusely via mossy fibers granule cell parallel fibers. The cortex processes this, with Purkinje cells integrating inputs and lateral inhibition sharpening the pattern. Finally, the Purkinje cell sends a precisely timed inhibitory signal back to the deep nucleus. The output of the deep nucleus, modulated by this excitation-inhibition balance, is the final signal sent out.
That's the essence of it.
Now let's connect this detailed circuitry back to that concept of the delay line damping mechanism, especially for preventing overshoot in rapid movements. How does this circuit achieve that?
This circuitry seems beautifully designed for exactly that function. Let's walk through a rapid movement scenario, say reaching quickly to touch a specific point. The command originates in the cerebral cortex or brainstem. This command information arrives at the cerebellum mainly via mossy fibers. These mossy fibers, remember, send collateral branches that directly excite the deep nuclear cells. This direct excitation provides a rapid, strong initial push or turn-on signal from the deep nucleus out to the motor pathways. The sources speculate this might even add extra force or accelerate the start of the movement initiated by the cortex.
The cerebellum gives the starting gun an extra kick.
Potentially, yes. Helps overcome inertia quickly. Now, simultaneously, those same mossy fibers are activating the pathway up into the cerebellar cortex. Mossy fiber excites granule cell fibers, sends action potentials along parallel fibers.
The indirect pathway.
Right.
Yeah.
And this pathway through the cortex involves multiple synapses and, importantly, those very thin, relatively slow-conducting parallel fibers. So there's an inherent physiological delay compared to the direct mossy fiber-to-deep nucleus connection.
The built-in delay line.
Exactly. After this brief but critical delay measured in milliseconds, the parallel fibers activate the Purkinje cells associated with this movement. The Purkinje cell integrates this input and fires. Since the Purkinje cell's output is inhibitory,
It sends a delayed inhibitory signal back down to the same deep nuclear cell that was initially excited and is currently driving the movement.
precisely. The deep nucleus gets the excitatory HO signal, and then, after a precisely calculated delay intrinsic to the cortical circuit, it gets the inhibitory SDOP signal from its Purkinje cell.
And that delayed stop signal is what prevents overshooting.
That's the crucial hypothesis. This delayed inhibitory feedback, arriving via the Purkinje cell, acts like a perfectly timed brake. It dampens the output of the deep nucleus, effectively helping to terminate the agonist muscle contraction just as the limb reaches the intended target. Without this precise predictive braking signal, the momentum of the rapid movement would carry the limb past the target,
Leading to oscillation.
leading to oscillation as you try to correct back, you'd overshoot, correct back, overshoot again. This is believed to be the underlying mechanism of intention tremor or action tremor, that classic cerebellar sign. The tremor occurs during the voluntary movement because the damping mechanism has failed.
Wow. Intention tremor really clicks into place now, seeing it as a failure of this specific delay line circuit and the Purkinje inhibition.
It provides a very compelling circuit-level explanation for a major clinical sign. The sources also speculate a bit further on the details of turn-on, turn-off timing for both the agonist muscles doing the work and the antagonist muscles opposing the movement.
How might that work?
Well, the rapid excitation from mossy fibers directly to deep nuclei could enhance the cortical turn-on signal for the agonists. Then, the delayed inhibition via the mossy granule parallel Purkinje pathway provides the timed turn-off signal for those same agonists. For the antagonist muscles, which need to relax at the start and then contract to help break the movement, the control likely involves both reciprocal inhibitory circuits within the spinal cord, automatically activated when agonists contract, and possibly other cerebellar circuits, perhaps involving inhibitory interneurons influencing the antagonist motor commands. The exact interplay is complex and still being researched, but the core agonist turn-on, turn-off timing seems well explained by the main cerebellar circuit.
It's an incredibly sophisticated biological timing device operating at the millisecond scale. And you mentioned earlier that this system, this precise timing and damping ability is learned. It's not just hardwired.
Absolutely critical point. The sources emphasize that the effectiveness of this cerebellar support the precise timing of the turn-on, turn-off signals. The strength of the damping, the coordination between muscles, is not innate. It is acquired and refined through practice and experience.
So when you first learn a new skill, like playing tennis or skiing, you're clumsy, movements are poorly timed, maybe overshoot.
Exactly. You might hit the ball too late, or swing too hard, or your skis cross. Movements are initially imprecise. With repetition, sensory feedback about the errors allows the cerebellum to adjust its internal workings, and performance gradually improves, becoming smoother, more accurate, and seemingly effortless.
How does that learning actually happen at the cellular or synaptic level within the cerebellum?
The prevailing theory, strongly supported by experimental evidence mentioned in the sourcing, is that motor learning involves long-term changes in the synaptic efficacy or sensitivity within the cerebellar circuits. Specifically, it's thought to involve changes in how effectively the Purkinje cells are excited by the inputs they receive from the parallel fibers.
Modifying the strength of those thousands of weak parallel fiber synapses.
Yes. And this modification process, this synaptic plasticity, is believed to be critically guided by the activity of the climbing fibers.
Ah, the climbing fibers again. The ones from the inferior olive that cause that big complex spike. How are they involved in learning?
The sources propose that the climbing fibers act as the crucial error signal that drives this motor learning process. Remember, they fire relatively infrequently, maybe once per second on average, but when they do fire, they cause that massive complex spike in the Purkinje cell.
So how do they signal an error? What tells the inferior olive there was an error?
When you perform a movement, especially a novel one, and it's not quite right, maybe you missed the target or the force is wrong, the sensory feedback systems detect this discrepancy between the intended movement and the actual outcome. This error information is relayed through various pathways we discussed to the inferior olive.
Okay, the olive gets the error report.
The inferior olive then adjusts the firing pattern of its climbing fibers accordingly. The sources suggest that when a complex spike, signaled by the climbing fiber, occurs in a Purkinje cell around the same time that specific parallel fibers are active, representing the context or components of the faulty movement, it triggers long-lasting changes in the strength of those particular parallel fiber to Purkinje cell synapses. Usually this is thought to be a long-term depression, LTD, a weakening of those synapses.
So the climbing fiber essentially says, whatever pattern of parallel fiber activity just happened when I fired, that pattern led to an error. Let's make that pattern less effective at driving the Purkinje cell next time.
That's the core idea of the leading theory, often called the Mar-Albus-Ito theory of cerebellar learning. The climbing fiber acts as a teacher, signaling when an error occurred, and this signal modifies the synaptic weights of the parallel fibers that were active just before or during the error. Over many repetitions, this selectively weakens the connections responsible for the erroneous part of the movement.
And this adjustment gradually refines the Purkinje cell's response.
Exactly. It changes how the Purkinje cell responds to future patterns of parallel fiber input associated with that movement. This, in turn, modifies the timing and pattern of the Purkinje cell's inhibitory output to the deep nucleus. The result is a gradual refinement of the deep nucleus output signal. leading to more accurate timing, better damping, and improved coordination until the movement becomes smooth and precise, conforming closely to the original intention.
And when the movement becomes perfect, does the climbing fiber stop signaling error?
Presumably, yes. Once the movement is consistently accurate, the sensory feedback indicates no error. The inferior olive would reduce or cease sending that specific error-related climbing fiber signal, and the synaptic weights would stabilize. The skill has been learned and stored, in part, within the adjusted sensitivities of the cerebellar circuits.
It's like the inferior olive, acting via the climbing fibers, is providing supervised learning for the cerebellum. Nope, that was wrong. Adjust. Nope, still wrong. Adjust again. Huh, that was a good. Keep it like that. Truly remarkable plasticity.
It really is a sophisticated mechanism for adaptive motor control. Now, with this understanding of the microcircuitry and learning, let's zoom back out again and look at how the cerebellum fits into the overall motor control system at different functional levels as the sources lay it out.
Right. The sources describe the cerebellum's role at three main levels: the vestibulocerebellum, the spinocerebellum, and the cerebrocerebellum. Let's start with the vestibulocerebellum.
Okay. This level corresponds primarily to those floccular and nodular lobes we mentioned earlier, plus adjacent parts of the vermis. As the name strongly implies, its function is deeply intertwined with the vestibular system. It's crucial for regulating body equilibrium and coordinating postural adjustments, especially during movement. It works in constant, close communication with the vestibular nuclei in the brainstem and the spinal cord pathways involved in posture.
How does it handle tricky balance situations like quick turns or sudden stops? That seems like a big challenge for feedback control because of delays.
That's exactly where the vestibulocerebellum shines. Maintaining balance is particularly difficult during rapid movements or changes in orientation because, as the sources point out, the sensory feedback signals from your feet or body position sensors can take a significant amount of time, maybe 15 to 20 milliseconds or more, to travel up the spinal cord and reach the brain. By the time that feedback arrives, your body might already be in a different position, especially if you're moving fast.
So simple feedback correction would be too slow.
It would often be too late to prevent instability. The vestibulocerebellum gets around this by using the very rapid signals coming directly from the vestibular apparatus in the inner ear. These signals report on head acceleration and orientation almost instantaneously. The vestibulocerebellum uses this immediate vestibular information to predict in advance where the different body parts are likely to be in the next few milliseconds.
It's doing predictive modeling based on head movement. It doesn't wait for the confirmation from the feet. It anticipates where the body will be and sends corrective postural signals based on that prediction.
That's the key insight. It makes anticipatory corrections to postural muscle activity based on predicting future body positions during motion. This feed-forward control, driven by vestibular input and internal models, is absolutely crucial for maintaining equilibrium during dynamic activities like running, jumping, or rapidly turning. It corrects for potential instability before it actually happens.
Predictive balance control. That's fascinating. Okay, the second level, the spinocerebellum.
The spinocerebellum comprises most of the vermis, excluding the parts associated with the vestibulocerebellum, and those intermediate zones on either side of it, within the anterior and posterior lobes. This level provides the circuitry for coordinating the ongoing execution of movements, particularly the movements of the distal portions of the limbs again. Think hands and fingers, feet and toes.
So coordinating the fine movements of our extremities as they happen, how does it achieve that coordination?
It functions primarily as a feedback control system, working in very close association with the brainstem and the cerebral motor cortex. Remember how the intermediate zones receive input? They get information about the intended movement plan descending from the motor cortex and the red nucleus, and they get the crucial real-time feedback about the actual movement execution from the peripheral proprioceptors via the dorsal spinocerebellar tract, and that efference copy of the motor command sent to the spinal cord via the ventral spinocerebellar tract.
Right, the plan, the result, and the command sent. It compares all three.
Exactly. Based on this continuous comparison, the output nuclei of this region, the interposed nuclei, generate corrective signals. These signals are sent back up to the cerebral motor cortex via the thalamus and also to the red nucleus, influencing the spinal tract. This feedback loop constantly adjusts the descending motor commands to ensure that the actual movements of the limbs, especially the agonist-antagonist muscle interplay, remain smooth, accurate, and coordinated with the overall plan.
And is this spinocerebellar level where that critical damping function preventing overshoot primarily resides?
Absolutely. This feedback loop involving the spinocerebellum intermediate zones, and interposed nuclei, is considered the primary system responsible for damping movements, particularly of the limbs, to prevent oscillation and ensure movements stop accurately at the target. Its failure is the main reason for intention tremor in the limbs.
Makes sense. The sources also say it's essential for ballistic movements. What are those exactly?
Ballistic movements are extremely rapid movements that are essentially pre-programmed and executed as a single unit. Think of swinging a bat, throwing a ball, or even the rapid saccadic eye movements we make to shift our gaze, or individual keystrokes when typing quickly. These movements happen so fast, often completed in fractions of a second, that there's simply no time for ongoing sensory feedback from the periphery to influence the movement during its execution.
They have to be planned entirely in advance, like firing a bullet.
Exactly like that. The entire trajectory, force profile, and timing have to be pre-calculated by the motor system. The sources emphasize that the cerebellum, particularly the spinocerebellum circuitry, is critical for executing these ballistic movements effectively.
How so? What does it contribute?
It seems to provide several key things. First, it likely helps generate the powerful initial burst of agonist muscle activity needed to launch the movement rapidly. Second, and perhaps even more critically, it provides the precise timing for turning off the agonist contraction and turning on the antagonist contraction at just the right moment to stop the movement accurately at the intended endpoint.
That precise on-off timing again?
Yes. Without the cerebellum, the sources state, these ballistic movements become severely impaired. They are slow to initiate, the force generated is often too weak, and crucially, the turn-off signal is delayed, causing the movement to overshoot the target significantly. The automaticity, speed, and accuracy of ballistic actions are lost. The cerebellum's internal timing circuits appear fundamental for this capability.
It adds the necessary punch to start and the crucial brake to stop these pre-planned rapid actions. Okay, that leaves the third functional level, the cerebrocerebellum.
The cerebellum corresponds to those large lateral zones of the cerebellar hemispheres, the parts that are most significantly expanded in primates, especially humans. The sources directly link this expansion to our highly developed abilities for planning, sequencing, and executing intricate, complex patterns of movement, particularly involving the hands and fingers, and also for complex skills like articulate speech.
And we established earlier that these lateral zones primarily communicate back and forth with the cerebral cortex, especially the planning and association areas.
Correct. Their input comes almost exclusively from the cerebral cortex, the premotor cortex, supplementary motor cortex, and various sensory association areas. Their output, via the dentate nucleus and the thalamus, loops primarily back to these same regions of the cerebral cortex. It forms a major cortex-cerebellum-cortex circuit dedicated to higher-level motor processing.
So what are the specific functions attributed to this cerebrocerebellar loop?
The sources identify two main closely related roles. First, it's heavily involved in the planning of complex sequential movements. This likely involves extensive two-way dialogue between the cortical planning areas and the lateral cerebellum. The initial idea or goal might arise in the cortex, which then communicates with the cerebellum to help work out the detailed motor strategy and sequence required.
And the second role?
The second role is providing the appropriate timing signals for these sequential movements. This timing function is critical. The lateral zones, leveraging their sophisticated internal timing circuits, perhaps similar in principle to the damping circuits, help the cortex determine the precise onset and termination times for each successive movement within a complex sequence. This ensures that the individual movements flow together smoothly and cohesively.
So it's not just planning what movements to make in sequence, but precisely when each one should start and stop relative to the others.
Exactly. The sources even note that neurophysiological recordings show activity in the dentate nuclei, the output of the lateral zones, often preceding the actual execution of the next movement in a planned sequence, sometimes by a significant fraction of a second. This strongly supports its role in anticipatory planning and timing, looking ahead to structure the upcoming sequence.
This really helps explain those failure of progression symptoms seen with cerebellar damage, doesn't it?
It absolutely does. Without the proper functioning of the lateral zones and their timing capabilities, the brain loses its subconscious ability to accurately predict how far different body parts will move during a given time interval. This makes it extremely difficult to smoothly coordinate the start and end times of successive movements in a rapid sequence, leading to movements that lack fluid progression. They become decomposed, jerky, and inefficient. This manifests clinically as dysdiadochokinesia, the inability to perform rapid alternating movements, like quickly flipping your hand back and forth. The movements become jumbled, irregular, and tend to stall. It also underlies dysarthria, the cerebellar speech disorder, where the lack of precise timing and coordination of the muscles involved in articulation, phonation, and respiration leads to speech that is slow, slurred, scanning, with unnatural separation of syllables and variable in volume. The smooth flow of speech breaks down.
The cerebellum as the master timer for movement sequences. The sources also briefly hinted at possible extramotor predictive functions for the cerebellum, something beyond just movement control.
Yes, it's a fascinating area of ongoing research. The suggestion is that these large lateral lobes, because of their connections with cortical association areas involved in processing sensory information, might also play a role in timing other rapidly changing events in the environment. For example, predicting the rate of approach of a moving object based on how quickly its image is changing on the retina, or interpreting complex, rapidly evolving auditory patterns.
So maybe predicting trajectories or rhythms in the outside world?
Possibly. The sources mention studies where monkeys with damage to the lateral cerebellum showed deficits in predicting trajectories and might misjudge distances, sometimes colliding with objects. This hints that the cerebellum's computational power for timing and prediction might not be strictly limited to motor control, but could provide a more general time-based or predictive capability used for interpreting rapidly changing spatiotemporal sensory information as well.
That's a really thought-provoking idea, the cerebellum as a kind of general-purpose prediction machine for rapid events, whether motor or sensory.
It definitely broadens the perspective on its potential functions. However, the sources are also careful to note that significant, lasting clinical dysfunction usually requires damage that involves the deep cerebellar nuclei. Small lesions confined just to the cerebellar cortex, especially in the lateral zones, might sometimes be compensated for, particularly if movements are performed slowly.
So while the cortex is doing the complex processing, the deep nuclei, the dentate, interposed, and fastigial are the critical choke points for the output. Damage there is what causes the most severe problems.
That seems to be the clinical reality, yes. They are the final common pathway for cerebellar influence on the rest of the motor system.
Okay, let's quickly recap the major clinical abnormalities that result from cerebellar damage, pulling it all together.
The absolute core symptoms are dysmetria, which is the inability to accurately judge the distance or range of a movement, leading to overshooting or undershooting the target, and the resulting ataxia, which is the general term for the lack of coordination and clumsiness of voluntary movements. This can also be made worse if the spinocerebellar tracts carrying sensory feedback are damaged.
And past pointing, where you reach past the target, is a specific manifestation of dysmetria.
Yes, often demonstrated by having the patient try to touch the examiner's finger and repeatedly going past it. It reflects the failure of the cerebellum to initiate the turn-off or braking signal at the right time.
Then there's the failure of progression, which encompasses the problems with sequencing and timing rapid movements. This shows up as dysdiadochokinesia, that difficulty with rapid alternating movements.
The hand flipping test, yeah.
And dysarthria, the uncoordinated, slurred, poorly timed speech.
Correct. We also see cerebellar nystagmus, which is an involuntary tremor or jerking of the eyeballs, especially when trying to fixate gaze on an object off to the side. This is seen as a failure of the damping mechanism for eye movements, and it's particularly associated with damage to the flocculonodular lobe and its connections related to equilibrium.
And finally, hypotonia.
Yes, hypotonia, which is a decrease in the normal background muscle tone observed on the same side of the body as the cerebellar lesion. Since cerebellar influences on the cortex are largely contralateral, but the cortex controls the opposite side of the body, the final effect is ipsilateral. This is thought to result from the loss of the normal tonic, facilitatory signals that the deep cerebellar nuclei continuously send out to the motor cortex and brainstem motor nuclei. Losing that background excitation leads to reduced muscle tone.
So, the cerebellum, far from silent, it's this indispensable, constantly active learned system for mastering the timing, damping, sequencing, and coordination of movements, particularly those that require speed, precision, and adaptation. Its damage truly reveals the hidden layers of complexity it normally adds to our motor control.
Absolutely. It's a remarkable structure. That pretty much wraps up our deep dive into the cerebellum based on the source material. Shall we now transition to the other major accessory motor system discussed? The basal ganglia.
Yes, let's move on to the basal ganglia, also described as an accessory motor system, also functioning in very close association with the cerebral cortex and the main corticospinal motor pathway. The sources really stress this tight connection. The basal ganglia receive most of their input from the cerebral cortex, and they send almost all of their output back to the cerebral cortex.
Exactly, highlighting their fundamental role as part of complex cortical processing loops, rather than having direct major outputs down the spinal cord like the motor cortex itself.
Okay, first, what structures actually make up the basal ganglia? The sources list several key components.
The main anatomical structures traditionally grouped under this term are the caudate nucleus, the putamen, and the globus pallidus, which itself has an internal and external segment. Functionally, two other structures are intimately linked and always discussed with them: the substantia nigra, located in the midbrain, and the subthalamic nucleus, located below the thalamus.
And where are these structures located?
They're situated deep within the cerebral hemispheres, mostly lateral to and surrounding the thalamus. The sources also make a point of mentioning the internal capsule.
The big fiber bundle.
Yes, the massive bundle of nerve fibers, including the corticospinal tract, connecting the cerebral cortex with the brainstem and spinal cord. This internal capsule actually passes between some of the basal ganglia structures, specifically separating the caudate nucleus and thalamus medially from the putamen and globus pallidus laterally. This close anatomical proximity underscores their intimate functional relationship with the major input and output pathways of the cerebral cortex.
Okay, now the circuitry within the basal ganglia is notoriously complex, but the sources helpfully divide their function based on two main, though interconnected, circuits: the putamen circuit and the caudate circuit. Let's tackle the putamen circuit first. What's its main role?
The putamen circuit is identified in the sources as being crucially involved in controlling complex patterns of motor activity that have become learned and largely automatic through practice. These are often movements we execute subconsciously once we've mastered the skill.
Can you give some examples from the sources of activities that rely heavily on this circuit?
Sure. The examples given include things like writing letters or words, cutting along a line with scissors, hammering a nail smoothly, throwing a baseball accurately, shooting a basketball, shoveling dirt efficiently. Most of the complex coordinated movements involved in vocalization and speech articulation and the fine control of eye movements. Basically, almost any skilled motor sequence that becomes relatively automatic and stereotyped with practice seems to depend on this putamen circuit operating correctly.
So it's involved in executing these learned motor programs or routines.
That seems to be the core function attributed to it, yes. Helping the cortex to run these complex multi-step routines smoothly and efficiently. The sources note that when this part of the basal ganglia is severely damaged, these learned patterns often lose their skillfulness and regress towards being very crude and deliberate, almost like how a beginner would perform them. For instance, handwriting might become slow, effortful, and poorly formed again.
Okay, so it helps execute learned skills.
Mhm.
Let's trace the main neural pathways involved in this putamen circuit. Where does the input come from?
The input to this circuit originates mainly from those parts of the cerebral cortex that are closely associated with motor function, but not primarily the primary motor cortex itself. Instead, it comes largely from the premotor cortex, the supplementary motor area, and also the somatosensory cortex in the parietal lobe, the parts closely related to the motor cortex.
So areas involved in planning movements and processing the sensory feedback related to those movements.
Exactly. These cortical areas project primarily to the putamen. In this circuit, the input largely bypasses the caudate nucleus. From the putamen, the signals travel to the internal segment of the globus pallidus, GPI. The GPI is one of the main output nuclei of the basal ganglia.
Okay, cortex, premotor, supplementary, somatosensory, putamen, internal globus pallidus. Where does the output from the GPI go?
From the GPI, the signals relay through specific nuclei in the thalamus again, primarily the ventroanterior, VA, and ventrolateral VL nuclei. And here's the crucial part of the loop. From the thalamus, these signals are projected back up primarily to the cerebral primary motor cortex, as well as returning to the premotor and supplementary motor areas.
Ah, so the loop closes by feeding back to the main execution area, the primary motor cortex, as well as the planning areas it started from.
Yes, it creates this loop. Input from perimotor planning sensory areas, processing through the putamen and globus pallidus, and output relayed via the thalamus back mainly to the primary motor cortex and associated areas to influence the execution of movements. The sources also mention that there are important ancillary or parallel circuits involving the external globus pallidus, the subthalamic nucleus, and the substantia nigra that modulate the flow through this main putamen GP-thalamus cortex loop.
And damage to different components within this complex circuitry is what leads to some of those specific movement disorders we hear about.
It provides a vivid illustration of how different nodes contribute. For example, lesions, specifically in the globus pallidus, are often associated with ethetosis, those slow, continuous, writhing, involuntary movements, particularly seen in the hands, arms, neck, or face.
Okay.
Damage to the subthalamic nucleus, which plays a key role in modulating the output of the globus pallidus, can result in hemibolismus characterized by sudden, wild, flailing movements of an entire limb on one side of the body. Very. And multiple small lesions, perhaps due to strokes, specifically within the putamen itself, can lead to chorea. Those more rapid, jerky, involuntary flicking or dancing movements, often seen in the hands, face, and other parts.
And the link to Parkinson's disease also falls under this putamen circuit's dysfunction.
Yes, absolutely. Profound damage to the substantia nigra, specifically the part called pars compacta, which provides critical dopamine input to the putamen and caudate, is the underlying cause of Parkinson's disease. This disruption leads to the characteristic symptoms of rigidity, tremor, and severe difficulty initiating movement, akinesia, which reflect a major breakdown in the smooth operation of this motor execution circuit.
It really highlights how disruption at different points in the circuit dedicated to executing learned patterns can lead either to an inability to perform movements smoothly, like Parkinson's, or the intrusion of unwanted involuntary movements, like ethetosis, hemibolismus, chorea.
Exactly. It underscores the circuit's role in both enabling desired patterns and suppressing unwanted ones. Now, let's contrast this with the caudate circuit. The sources associate this second major circuit more with the cognitive control of motor patterns.
Cognitive control. What exactly does that mean in the context of movement?
The sources define cognition generally as the thinking processes of the brain. Most of our purposeful actions, our movements, don't just happen randomly. They are initiated as a consequence of our thoughts, intentions, goals, and our perception of the environment. The caudate circuit is described as playing a major role in selecting and sequencing motor patterns appropriately in order to achieve a complex goal that originates from these cognitive processes.
Okay, so if the putamen circuit is more about the smooth execution of already learned, somewhat automatic patterns, the caudate circuit is more involved in the higher-level decision-making about which patterns to use in what order and when, based on our thoughts and the current situation.
That's the key distinction the sources seem to be making, yes. The caudate circuit helps translate cognitive intent into appropriate motor strategies. It helps us perform complex actions that are a direct consequence of conscious thought processes rather than just running stereotyped subroutines.
How are its neural connections different from the putamen circuit to support this role?
Well, structurally, the caudate nucleus is different. It's this large C-shaped structure that follows the curve of the lateral ventricle and extends throughout all the lobes of the cerebral cortex. Because of this extensive reach, it receives massive input from the widespread association areas of the cerebral cortex.
Not just the motor-related area.
No, much broader input from areas involved in integrating different types of sensory information, linking information to memory, spatial processing, decision-making, executive functions, essentially. The parts of the cortex most involved in higher-level thought processes, which often physically overlie the caudate nucleus in different lobes.
So its input comes directly from the brain's thinking and integrating regions. Where do the signals go from the caudate?
From the caudate nucleus, signals travel, similar to the other circuit, to the internal globus pallidus, GPI, the main output structure. From the GPI, they relay through the VA and VL nuclei of the thalamus. But here's the critical difference in the output target. The signals return primarily to the prefrontal cortex, the premotor cortex, and the supplementary motor areas.
Noticeably different again. The output goes back predominantly to these higher-level planning and executive control areas of the cortex, not primarily to the primary motor cortex itself, like the putamen circuit did.
That's a key distinction highlighted in the sources. They state that almost none of the output from this caudate circuit goes directly back to the primary motor cortex. Instead, by influencing these prefrontal, premotor, and supplementary motor areas, the regions concerned with motor planning and strategy, the caudate circuit is thought to play a role in selecting and sequencing patterns of movement over longer timescales, perhaps patterns lasting 5 seconds or more, rather than directly exciting individual muscles.
So it's operating at a more strategic level, influencing the assembly of complex, goal-directed action sequences.
That seems to be the idea. It helps determine which combination of fundamental or learned motor patterns needs to be activated, and in what sequence to achieve a complex behavioral goal that originated from a thought process. The sources use the example of seeing a predator, like a lion.
Right.
The thought process is danger, escape. This cognitive assessment, likely involving association cortex input to the caudate, then needs to rapidly trigger an appropriate sequence of actions. Maybe turn, run, look for a tree, climb the tree. The caudate circuit is proposed to help select these necessary motor patterns, turning, running, climbing, likely executed via the putamen circuit or brainstem, and initiate their rapid subconscious sequence based on that initial cognitive appraisal. It bridges the gap between the thought, danger, and the complex motor plan needed to respond effectively.
That makes a lot of sense. The caudate circuit acts as an interface, translating high-level cognitive goals and situational assessments into the selection and sequencing of appropriate motor strategies, which are then implemented by other parts of the motor system.
It provides that cognitive dimension to motor control. The basal ganglia as a whole are also described as contributing significantly to changing the timing and scaling of movements.
Timing and scaling. Can you elaborate on that?
Yes. Timing here refers partly to determining how quickly a movement should occur. Scaling refers to controlling its amplitude or overall size. The source uses the excellent example of writing the letter A. You can write a tiny A very quickly on a piece of paper, or you can write a huge A slowly on a whiteboard.
Right, the shape is recognizably the same, but the speed and size are completely different.
Exactly. The relative proportions and timing characteristics of the muscle contractions involved remain remarkably similar across these different scales and speeds. The basal ganglia are thought to play a crucial role in managing this ability to scale movements in both time and space while preserving their essential form.
And is this ability impaired in patients with basal ganglia disorders?
Yes, the sources state that in patients with severe basal ganglia lesions, these functions of timing the speed of movement and scaling the amplitude of movement are often markedly impaired or even lost. Movements might become abnormally slow, bradykinesia, or inappropriately small, micrographia, as seen in Parkinson's.
And like the cerebellum, the basal ganglia don't do this in isolation, right? They work closely with the cortex.
Absolutely. The source specifically highlights the close functional relationship between the basal ganglia and the posterior parietal cortex. This region of the cortex is known to be critical for constructing our sense of spatial coordinates, maintaining our body schema, our internal map of our body, and understanding the relationship between our body and the surrounding environment.
Okay, posterior parietal cortex for spatial awareness. What happens if that cortical area is damaged?
Damage there typically doesn't cause simple loss of sensation or paralysis. Instead, it often results in complex deficits known as agnosias, an inability to perceive or recognize objects or spatial relationships correctly, even though the basic senses might be intact. The source gives the example of a patient with damage to the right posterior parietal cortex who might be asked to copy a drawing of a house or a clock. They might only draw the right side of the object, completely neglecting the left side as if it doesn't exist in their spatial awareness.
Wow, neglecting half of space.
Yes. Related to this is the striking condition called personal neglect syndrome, where the individual might ignore one entire side of their own body. They might fail to dress that side, wash it, or even acknowledge that it belongs to them. It vividly illustrates how crucial this cortical area is for our internal representation of body space and its relation to external space.
So how does this connect back to the basal ganglia and timing scaling?
The sources speculate, and they emphasize this is more analytical deduction than proven fact. That because the caudate circuit has such extensive connections with these cortical association areas, including the posterior parietal cortex, the functions of timing movement speed and scaling movement amplitude might be mediated, at least in part, through this cognitive control loop. The caudate circuit, by interacting with areas that define spatial parameters, could help set the appropriate scale and timing for intended actions.
So the cognitive circuit might be leveraging spatial information from the parietal cortex to help determine how large or fast a movement should be.
That's the plausible hypothesis put forward. acknowledging that the precise mechanisms are still under investigation. But the clinical evidence strongly implicates the basal ganglia in these crucial aspects of tailoring movements to specific contexts.
Okay, now let's talk about the neurochemistry. The basal ganglia seem to be a real hub for complex interactions between different neurotransmitters. Getting that balance right seems absolutely critical.
It is absolutely crucial. The sources highlight several key transmitter systems. One of the most famous involves dopamine. Neurons originating in the substantia nigra, specifically the pars compacta, send axons that release dopamine in the caudate nucleus and putamen. Dopamine's effects in the basal ganglia are complex, acting on different receptor subtypes, but overall it's considered to be largely inhibitory within the main circuits and acts as a critical modulator or stabilizer for motor control loops. Its loss is, of course, the cause of Parkinson's disease.
Okay, dopamine as a key modulator, mostly inhibitory. What about GABA? That's gamma aminobutyric acid.
GABA is the main inhibitory neurotransmitter used by many neurons within the basal ganglia. For example, neurons projecting from the caudate and putamen down to the globus pallidus and the substantia nigra, pars reticulata, another output nucleus, release GABA. The sources make the important point that the vast majority of the feedback loops from the basal ganglia back to the cortex are effectively negative feedback loops, and this inherent stability relies heavily on the inhibitory influence of these GABAergic pathways.
So GABA provides much of the braking within the system. What about excitatory influences?
Glutamate is the primary excitatory neurotransmitter in the brain, and it plays a key role here too. The massive input pathways from the cerebral cortex to the caudate and putamen use glutamate, providing the main excitatory drive into the basal ganglia. The pathway from the subthalamic nucleus to the globus pallidus is also glutamatergic and excitatory. So there's a constant interplay between glutamate excitation and GABA and dopamine inhibition.
And there are other neurotransmitters involved too.
Yes. Acetylcholine, ACMA, is also important. There are large acetylcholine-releasing interneurons within the caudate and putamen themselves, which play a complex modulatory role, often acting in opposition to dopamine. There are also pathways originating in the cortex that release acetylcholine onto basal ganglia neurons. Additionally, the sources mention pathways from the brainstem that release other transmitters like norepinephrine, serotonin, and various neuropeptides like enkephalin within the basal ganglia, adding further layers of modulation.
It sounds like an incredibly complex chemical balancing act.
It really is. The normal, smooth functioning of the basal ganglia depends entirely on the precise balance between these multiple excitatory and inhibitory neurotransmitter systems. Disrupt this delicate neurochemical equilibrium either by losing an inhibitory influence like dopamine in Parkinson's or losing GABA inhibition in Huntington's, and you get the dramatic and debilitating motor syndromes that clinically define basal ganglia dysfunction.
Which brings us neatly back to summarizing those major clinical syndromes resulting from basal ganglia damage. We already touched upon ethetosis, linked to globus pallidus, hemibolismus, subthalamus, and chorea putamen, as examples of specific lesion effects. But the two major diseases highlighted are Parkinson's and Huntington's. Let's revisit Parkinson's disease first, also known as paralysis agitans.
Right. Arguably the most widely recognized basal ganglia disorder.
And the underlying cause, according to the sources.
It's unequivocally caused by the progressive degeneration and death of the dopamine-secreting neurons located in the pars compacta region of the substantia nigra. These are the neurons that provide the crucial dopamine innervation to the caudate nucleus and the putamen, collectively often called the striatum.
So, a devastating loss of dopamine input to the main input structures of the basal ganglia. What are the classic hallmark symptoms that result?
The sources list three cardinal motor features. First, significant rigidity of the muscles throughout much of the body, often described as lead pipe or cogwheel rigidity. Second, an involuntary tremor, which is characteristically most prominent at rest, the so-called pill-rolling tremor, typically occurring at a frequency of about 3 to 6 cycles per second. Third, and often the most disabling symptom, is severe akinesia or bradykinesia.
Akinesia meaning difficulty initiating movement, and bradykinesia meaning slowness of movement.
Exactly. Akinesia is the profound difficulty in getting movement started, a freezing or poverty of spontaneous movement. Bradykinesia is the marked slowness once movement is initiated. Patients often describe needing immense conscious mental effort just to begin a simple action, like standing up from a chair.
That rest tremor is a key feature distinguishing it from cerebellar tremor, which occurs during action.
Yes, that's a very important clinical distinction. Cerebellar tremor is an intention or action tremor. Parkinsonian tremor is typically maximal at rest and often decreases during voluntary movement. Other common motor symptoms in Parkinson's include postural instability, impaired balance reflexes leading to frequent falls, usually later in the disease, difficulty with swallowing, dysphagia, soft or monotonous speech, hypophonia, a shuffling gait with reduced arm swing, and significant fatigue.
That akinesia sounds particularly difficult to live with.
It is often reported by patients as the most frustrating and limiting symptom. They describe feeling stuck or frozen, requiring tremendous willpower to overcome the inertia and initiate movement. When movements do occur, they lack normal fluidity, often being performed stiffly, hesitantly, and sometimes in a series of small, shuffling steps or increments.
How does the loss of dopamine actually lead to these specific symptoms based on the circuit understanding? Rigidity, for instance.
The prevailing hypothesis is that since dopamine normally exerts a net inhibitory or stabilizing influence on the neurons in the caudate and putamen, via complex effects on direct and indirect pathways, its loss leads to these structures becoming pathologically overactive or dysregulated. This altered activity propagates through the basal ganglia loops, ultimately resulting in excessive inhibitory output from the basal ganglia, from GPI and SNR, to the thalamus. Reduced thalamic excitation of the cortex then leads to decreased cortical drive, but also complex changes in downstream pathways are thought to contribute to the rigidity, perhaps by increasing activity in brainstem pathways that enhance muscle tone. The exact mechanisms are still debated, but involve disinhibition of certain pathways.
Okay, so loss of dopamine, overactive basal ganglia output, altered cortical and brainstem drive rigidity. What about the rest tremor?
The tremor is thought to arise from oscillatory rhythmic firing patterns emerging within the basal ganglia thalamocortical loops. Normally, dopamine helps to stabilize these loops and prevent synchronous oscillations. When dopamine is lost, certain feedback loops within the basal ganglia circuit may become unstable and start to oscillate at that characteristic 3 to 6 Hz frequency. This rhythmic activity is then transmitted through the thalamus to the motor cortex and down to the muscles, manifesting as the rest tremor.
And the akinesia, bradykinesia, the difficulty starting, and the slowness.
This is perhaps the least well understood symptom mechanistically. One leading hypothesis mentioned in the sources is that it relates to the basal ganglia's role in scaling movement amplitude and velocity. The loss of dopamine may impair the ability to generate sufficient internal drive or activation within the basal ganglia loops to initiate movements at the desired speed and amplitude. Another contributing factor, also speculated upon, might involve reduced dopamine levels not just in the motor circuits, caudate to putamen, but also in parts of the limbic system associated with motivation and reward, such as the nucleus accumbens, sometimes called the ventral striatum.
Ah, the motivational connection again?
Yes. Since the limbic system is crucial for generating the psychic drive or motivation to act, a reduction in dopamine signaling within these limbic loops could contribute to the apathy and lack of spontaneous movement initiation seen in akinesia. It might diminish the fundamental wanting to move.
The sources also mention common non-motor symptoms, like sleep disturbances, depression, anxiety, autonomic dysfunction like constipation or orthostatic hypotension, and cognitive impairment or dementia, particularly in later stages.
Yes, it's increasingly recognized as a multi-system disorder. The neurodegeneration is not strictly confined to the substantia nigra dopamine cells, but affects other brain regions and neurotransmitter systems over time, leading to this wide range of non-motor features which significantly impact quality of life.
What about treatments for Parkinson's mentioned in the sources? L-DOPA is the classic therapy.
L-DOPA, levodopa, remains the most effective symptomatic treatment. It's a precursor molecule that the brain can use to synthesize dopamine. Unlike dopamine itself, L-DOPA can cross the protective blood-brain barrier. Once inside the brain, the remaining dopamine neurons, and possibly other cells, convert L-DOPA into dopamine.
So it boosts the dwindling dopamine supply.
Exactly. It helps to replenish dopamine levels in the striatum, caudate and putamen, temporarily restoring a better balance between excitation and inhibition within the basal ganglia circuits. This typically provides significant relief from the motor symptoms, especially rigidity and bradykinesia, for many years. However, its effectiveness can decrease over time, and long-term use is often associated with complications like motor fluctuations, on-off periods, and dyskinesias, L-DOPA-induced involuntary movements. It also generally has less effect on the tremor and many of the non-motor symptoms.
Other drugs mentioned include monoamine oxidase inhibitors, MAOIs.
Yes, specifically MAOB inhibitors. These drugs work by inhibiting the enzyme, monoamine oxidase B, that breaks down dopamine after its release. By slowing dopamine degradation, they help to prolong the action of any dopamine that is still being released, or dopamine derived from L-DOPA. The sources also note there's some evidence suggesting these drugs might have a mild neuroprotective effect, possibly slowing the underlying progression of neuron loss, though the mechanism isn't fully established. They are often used in early stages or in combination with L-DOPA.
And looking towards the future, fetal dopamine cell transplantation is mentioned.
This is an experimental approach, where dopamine-producing cells from fetal brain tissue are surgically implanted into the striatum of Parkinson's patients. The sources note that some clinical trials have shown encouraging short-term results, with evidence of graft survival and some symptomatic improvement in certain patients. However, there have also been challenges, including variability in outcomes and the development of graft-induced dyskinesias in some cases. If researchers can overcome these hurdles and achieve consistent, long-term functional integration of the transplanted cells, it holds theoretical promise as a restorative therapy to replace the lost dopamine neurons, but it's not yet a standard clinical treatment.
Okay, that's a comprehensive overview of Parkinson's. Now let's turn to the other major condition discussed. Huntington's disease, sometimes called Huntington's chorea. Very different cause, very different symptoms.
Dramatically different, yes. Huntington's is a devastating autosomal dominant hereditary disorder. This means inheriting just one copy of the mutated gene from an affected parent is sufficient to cause the disease. Symptoms typically don't appear until adulthood, often manifesting between the ages of 30 and 40, though juvenile onset can occur.
And the symptoms are almost the opposite of Parkinson's akinesia, right? More about excessive movement.
Exactly. Instead of a poverty of movement, Huntington's is characterized by an excess of unwanted involuntary movements. It typically begins subtly, perhaps with restlessness or fidgetiness, and progresses to the development of chorea, those brief, irregular, rapid, dance-like involuntary movements that flit from one body part to another. As the disease advances, these choreic movements often become more severe and blend with slower, writhing movements, dystonia, and postural instability, leading to increasingly severe whole-body distortional movements that significantly impair function.
So uncontrolled, excessive movement. But there's another critical feature too.
Yes. Alongside the progressive motor disorder, Huntington's invariably involves profound cognitive decline and psychiatric disturbances. Patients develop severe dementia, with problems in executive function, memory, and judgment, as well as mood disorders like depression, anxiety, irritability, and sometimes psychosis. It's a combined motor, cognitive, and psychiatric illness.
What's the underlying neuropathology? What causes these specific symptoms?
The sources state that the primary pathology involves extensive neuronal loss, particularly in the caudate nucleus and putamen, the input structures of the basal ganglia. Specifically, there's a profound loss of the medium spiny neurons, which are the main projection neurons of these structures, and primarily use the inhibitory neurotransmitter GABA. There is also significant loss of acetylcholine secreting neurons, not just in the basal ganglia, but in many parts of the brain, including the cortex.
So a major loss of key inhibitory neurons within the basal ganglia, especially the GABAergic ones. How does that lead to the chorea?
The loss of these GABA neurons disrupts the normal balance of activity within the basal ganglia circuits. Specifically, the GABAergic projection from the striatum normally inhibits parts of the globus pallidus, particularly the external segment in the pathways leading to chorea, and the substantia nigra. When this inhibition is lost due to neuron death, these downstream structures become disinhibited or pathologically overactive. This increased, uncontrolled activity is then thought to drive excessive and inappropriate excitatory output from the thalamus back to the cortex, triggering the spontaneous involuntary choreic movements.
So loss of inhibition leads to overactivity and unwanted movements. What about the severe dementia?
The dementia is likely related to the widespread neuronal loss, particularly the significant loss of acetylcholine-releasing neurons, which are known to be crucial for cognitive functions like learning and memory. The degeneration extends beyond the basal ganglia to involve the cerebral cortex itself as the disease progresses, contributing further to the cognitive decline. The exact mechanisms linking the protein abnormality to dementia are still being worked out.
And the genetic cause is known.
Yes, the specific genetic defect was identified in 1993. It's an abnormal expansion of a repeating sequence of 3 DNA bases, CAG, within a gene located on chromosome 4. This gene codes for a protein called huntingtin. In healthy individuals, the CAG segment is repeated a relatively small number of times. In Huntington's patients, this segment is abnormally expanded, leading to the production of a mutated Huntington protein containing an elongated tract of the amino acid glutamine.
And this mutated protein is somehow toxic to neurons.
That's the central issue. It's clear that this mutated Huntington protein is toxic, particularly to those vulnerable GABAergic neurons in the striatum and acetylcholine neurons. But the exact mechanisms by which it causes cell dysfunction and death are incredibly complex and remain a major focus of intensive research. Understanding how the mutant protein disrupts cellular processes like protein folding, transport, transcription, and energy metabolism is key to developing effective therapies, which are currently lacking.
So, summarizing the contrast. Parkinson's involves primarily a loss of dopamine input, leading to downstream circuit changes that cause difficulty initiating movement, akinesia, bradykinesia, and tremor rigidity. Huntington's involves primarily a loss of GABAergic and cholinergic neurons within the basal ganglia, leading to disinhibition and excessive, uncontrolled movements, chorea, along with dementia.
That's a good distillation of the core differences based on the source material. Both diseases tragically illustrate the catastrophic consequences of disrupting the intricate neurochemical balance and circuit integrity within these crucial basal ganglia structures. They are powerful clinical windows into the fundamental role these nuclei play in enabling purposeful, stable motor control.
This has been an incredibly detailed exploration of the cerebellum and the basal ganglia individually. Now let's take a step back and try to bring it all together, looking at how they integrate into the total motor control system as the sources describe it.
Right. The sources paint a picture of motor control operating hierarchically across multiple levels of the nervous system. At the very lowest level, you have the spinal cord. It contains the basic, hardwired circuitry for fundamental reflexes like the stretch reflex or withdrawal reflex, and also contains more complex, intrinsic programs for rhythmic movements, like the basic patterns for walking or scratching. These spinal circuits can be activated, modulated, or even completely overridden by commands descending from higher brain centers.
Okay, spinal level for basic reflexes and patterns. Above that,
You have the hindbrain level, encompassing structures in the brainstem, like the medulla, pons, and midbrain. This level is crucial for maintaining basic axial muscle tone needed to support the body against gravity and for executing continuous postural adjustments, especially those related to equilibrium, working closely with the vestibular system.
and then the motor cortex level.
The cerebral motor cortex is seen as the primary executive level for initiating most voluntary movements. It can directly activate spinal cord motor neurons via the corticospinal tract, command the activation of those lower-level spinal cord patterns, or implement higher-level, complex, learned patterns of movement that are likely stored or organized within cortical circuits themselves. It has the ability to issue specific commands and bypass or modify lower-level functions.
So spinal cord with basic programs, hindbrain for posture tone, motor cortex for initiating voluntary action and commanding patterns. Where do the cerebellum and basal ganglia fit into this hierarchy? Are they separate levels?
The sources emphasize that they aren't really separate levels in the same way, but rather critical accessory systems that work in parallel with and modulate the activity of all these other levels. They provide essential processing loops that refine and enable the functions of the cortex, brainstem, and spinal cord.
Okay, let's look at the cerebellum's integration first. How does it interact across these levels?
As we've discussed in detail, the cerebellum provides crucial support, particularly for movements that require speed, precision, and adaptation based on sensory feedback. It interacts with the spinal cord level, for example, by modulating reflexes, like the stretch reflex, potentially enhancing its responsiveness when posture is challenged by unexpected loads.
Okay.
It works closely with the brainstem level, especially the vestibular cerebellum, to smooth out and coordinate the rapid postural and equilibrium adjustments needed during dynamic movement.
And its interaction with the cerebral cortex seems paramount.
Absolutely. The cerebrocerebellum, lateral zones, engages in that constant dialogue with the cortical planning areas to help plan, sequence, and time complex voluntary movements. The spinocerebellum interacts with the motor cortex and red nucleus to provide real-time feedback correction and damping for ongoing limb movements. The sources stress that the cerebellum is particularly critical when movements are rapid or when the intended motor plan needs to be changed quickly based on evolving sensory information.
So it's essential for the speed, accuracy, timing, and adaptability of voluntary movements orchestrated by the cortex.
Precisely. The sources highlight that while slow, deliberate movements might still be possible, albeit clumsily, without a functioning cerebellum, The ability to execute rapid, smooth, coordinated, and learned motor skills is profoundly compromised. Its functions timing, damping, coordination, learning are essential additions to the basic motor commands, primarily applied to ensuring the quality and precision of action, especially fast action. And these functions are largely learned and refined through experience.
Okay, that clarifies the cerebellum's integrative role. Now, how do the basal ganglia fit into this overall picture? How do they integrate?
The basal ganglia also function as essential accessory loops, primarily interacting with the cerebral cortex, but contributing in complementary yet distinct ways compared to the cerebellum. Their key integrative roles, based on the putamen and caudate circuits, seem to be twofold.
What are they?
First, they help the cerebral cortex to execute subconscious, learned motor patterns smoothly and automatically. Think of skills like writing, typing, playing an instrument, aspects of speech. This relates mainly to the putamen circuit. They essentially help run these well-practiced motor subroutines efficiently.
Okay, executing learned habits or skills. The second role.
Second, they are involved in the higher-level planning and sequencing of multiple motor patterns, both parallel and sequential, that the mind needs to string together to achieve a specific, purposeful, often complex task. This relates more to the caudate circuit. This includes helping to control the overall dimensions, like the timing, speed, and scaling amplitude of the movements within that plan.
So facilitating the execution of ingrained routines and also helping to select, sequence, and scale those routines into larger, goal-directed actions based on cognitive processes.
Yes, that captures the essence of their contribution as described. The sources particularly emphasize the role of the caudate circuit in providing that high-level interface between cognitive processes, thoughts, intentions, situational assessments originating in the association cortex, and the generation of appropriate, complex, sequential motor strategies needed to act on those cognitions. Like deciding to respond to a complex social cue or navigating a challenging environment based on a mental map.
So putting it together, while the cerebellum seems specialized for the real-time coordination, timing, damping, and error-based learning, particularly crucial for rapid and ballistic movements and postural stability,
Right.
the basal ganglia seem more specialized for the initiation, termination, sequencing, and scaling of movements, especially learned patterns, and those guided by cognitive goals and context, acting more like a selection and gain control mechanism for motor programs.
That's an excellent summary of their complementary roles. They are both indispensable accessory systems, neither causing movement directly in isolation, but both working intimately with the cerebral cortex, brainstem, and spinal cord. They each provide a unique set of computational services. The cerebellum focusing intensely on the how of movement execution, precise timing, coordination, adaptation, damping, while the basal ganglia focus more on the what, when, and how much, pattern selection, sequencing, initiation, scaling, linking to cognition. Together, their constant parallel processing ensures that our total motor output isn't just possible, but is skillful, adaptive, smooth, contextually appropriate, and purposeful.
This deep dive has really illuminated the incredible amount of sophisticated, largely subconscious processing that underpins our ability to simply move through the world effectively. It's quite humbling.
It truly is. It's a powerful reminder of the brain's astonishing complexity and the elegance of its integrated networks. These structures, the cerebellum and basal ganglia, operating mostly outside our conscious awareness, are constantly sifting through torrents of information, running complex calculations based on intricate circuits and precisely balanced neurochemistry, and sending out exquisitely refined signals to shape our every action, from the simplest gesture to the most complex skill.
Now, finally, the sources raise one more fundamental question at the very end. What is it that actually initiates movement in the first place? What provides the underlying drive for us to act at all?
Right, it touches briefly on the brain's motivational systems. These are primarily associated with what's sometimes called the older core of the brain, the limbic system. This system includes a collection of interconnected structures like the hypothalamus, the amygdala, the hippocampus, the septal region near the front, and parts of the older thalamic and cortical areas surrounding the brainstem.
And the limbic system's role is linked to providing the initial spark for action.
The sources state that these limbic structures, functioning together, are thought to be responsible for generating the motivational impetus, the psychic drive, that initiates most of the motor activities, as well as other functional activities, of the brain. They link our internal states, needs, emotions, goals, to the initiation of behavior.
And they connect this back to Parkinson's,
Yes, they reiterate the speculation that the severe akinesia in Parkinson's might be partly due to reduced dopamine function, not just in the motor basal ganglia, but also within key limbic structures like the nucleus accumbens, ventral striatum, which is heavily involved in motivation and reward processing. A deficit here could potentially diminish that fundamental drive or wanting to initiate movement, contributing to the feeling of apathy and effort required to start actions.
That really adds another crucial layer to the picture. Movement isn't just a mechanical output triggered by a neutral command. It's often, perhaps usually, an expression of our internal motivational state, our needs, desires, and emotions originating in these ancient limbic areas. These drives then recruit the sophisticated machinery of the cortex, basal ganglia, and cerebellum to translate that intent into effective, coordinated action in the world.
It beautifully highlights how the total motor control system is not just an isolated engineering system for moving muscles, but a deeply integrated network connecting sensation, perception, cognition, learning, refinement, execution, and even our most fundamental motivations, all working together to produce purposeful behavior.
This has been an absolutely fascinating journey into the hidden, intricate mechanics of our motor control systems. Thank you for guiding us through that complexity.
My pleasure. It certainly gives you a profound new appreciation for the neural marvels that allow us to interact with our world so seamlessly most of the time.
It really does. So let's leave you, our listeners, with this final thought to ponder. Consider the next complex, rapid movement you make, maybe something as automatic as catching a set of keys someone tosses you, or as intentional as quickly navigating a crowded sidewalk while checking your phone, or even just the complex, fluid coordination required to speak your thoughts rapidly and clearly. Reflect on the incredible, precisely timed subconscious interplay unfolding within your brain. Your cerebral cortex initiating the overall goal and strategy, your cerebellum instantly refining the timing, predicting trajectories, damping oscillations, and correcting for errors based on learned models and real-time feedback, and your basal ganglia helping to select and sequence the appropriate learned motor patterns, scaling them correctly based on your underlying cognitive goals and the environmental context. So much of this intricate, high-speed collaboration is learned, automatic, and happens far below the threshold of your conscious awareness. It makes you wonder what other seemingly simple, everyday human abilities, perhaps perception, language understanding, social interaction, might also rely on similarly complex, hidden, and subconscious teamwork between different specialized systems within the brain.
It's a question that reminds us just how much hidden computational sophistication underlies our experience of being human.
Absolutely. That's all the time we have for this deep dive. We hope you feel significantly more informed about these critical brain structures and perhaps have a renewed sense of awe for the incredible biological machinery operating within your own head.